Hello, Dr. Hudson. This is Megan Lahan. I'm part of your online cell biology course, and this is my presentation on one of the scientific journals that I chose. I initially started reading this one after I realized I was assigned DNA ligase. I had heard about something called DNA ligase 4 deficiency and how it could cause a wide variety of odd symptoms in humans, including skin discoloration and some odd abscesses on the flesh. And these kind of medical curiosities have always made me curious, so I wanted to investigate it further. So when I saw ligase 4 was mentioned in this paper, I read it. It did not in fact cover DNA ligase 4 deficiency, but I did find it interesting and relevant to my paper. The scientific journal was entitled DNA ligase 4 and Artemis Act Cooperatively to Suppress Homologous Recombination in Human Cells, Implications on DNA Double-Strand Break Repair. It was written by Kurosawa, Saito, So, Hashimoto, Iwabuchi, Watabe, and Adachi. The research and writing was done at the Graduate School of Nanobioscience, Yokohama City University in Yokohama, Japan. What they had done in this paper was, in their experiments obviously, was they were looking at the ratio of homologous recombination repairs to the amount of double-strand break repairs done by non-homologous end joining. In the researchers' opinion, homologous recombination was a safer, better way for double-stranded break repairs to occur. So why was it then that in their research they seemed to find that non-homologous end joining occurred more often than homologous recombination? So they toyed around with how they could get, with the different proteins involved with both processes and tried to see if they could alter the ratio in which the two occurred. So they isolated a human genome, and by using gene knockout techniques, they canceled out the sequence for 3 specific proteins: RAD54, DNA ligase 4, and Artemis. And in the first experiment, when they canceled out RAD54, they noticed homologous recombination dropped significantly. So when they looked into it further, they noticed that the protein responsible for homologous recombination was associating with a very specific protein filament called, or nucleofilament called RAD51. So what RAD54 was doing is it was combining with RAD51, and it was allowing that protein that carries out homologous recombination to more easily associate with RAD51. Once the two were associated and homologous recombination was occurring, RAD54 just kind of dropped off. They didn't say what happened to it, but I'm assuming it's just flitting about in the cytosol waiting to be used again. The second protein that they canceled the sequence out for was DNA ligase 4, specifically human DNA ligase 4. And what they noticed is that when they removed DNA ligase 4 from the equation, non-homologous end joining stopped completely. So it was assumed by the researchers that non-homologous end joining absolutely required DNA ligase 4 to be present in order to occur. So the third and final protein that the sequence they removed was for Artemis, something just simply called Artemis. I have no prior familiarity with Artemis, but what they noticed was that when they removed it, non-homologous end joining noticeably slowed. It didn't drop as far as homologous recombination did with the removal of RAD54, but they noticed that it did drop significantly. So it is assumed that Artemis is acting in cooperation with DNA ligase to make non-homologous end joining occur more frequently than homologous recombination. An interesting side effect they found was when Artemis was removed, the cells were also more resilient to low ionized radiation, which I would not fully understand the implications of, but find interesting nonetheless. I would like to use this in my paper to reinforce one of the points I'm going to make when I say that ligase is a necessity for life to occur in all organisms. Prior to reading this paper, I hadn't really explored the idea of DNA ligase being used in double-strand break repairing. I had primarily focused on how it was used in combining the Okazaki fragments during gene replication. So I would like to use this information from this article to kind of bolster my statement that DNA ligase is an absolutely necessary protein in all living creatures, not just humans, but in the various organisms that it exists within, rats, yeast, etc., and etc. So this was the one paper that I found the most interesting, and definitely I found it helpful for writing my paper. And I just wanted to share this with you in the form of this video. Thank you for watching. This is, again, Ben Megan Lahan, and this is my presentation on DNA ligase 4 and Artemis act cooperatively to suppress homologous recombination in human cells, implications for DNA double-strand break repair. Thank you.